Very good morning, everyone. Today I'm going to talk about plaque as a biofilm. So in this today, I will be talking about dental plaque as a biofilm. So in this, I'll be discussing about what is meant by dental plaque, how it is getting formed, what are the various theories, what is the classification, and the composition of dental plaque, what are the various physical properties of the dental plaque, neuron sensing, uh, and association of plaque with periodontal disease, criteria for identification of periodontal pathogens, and what are the various newer strategies for dental plaque formation. So dental plaque is defined as a specific and highly variable sequential colonization and growth of microorganisms on the surfaces of teeth, and the restoration consisting of microorganisms of various strains and species embedding in extracellular matrix, which is composed of bacterial metabolic products and substances from serum, saliva, and blood. It is also defined as a structured, resilient, yellow-grayish substances adhering tenaciously to the intraoral surfaces, including removable and fixed restorations. It should be differentiated from other two entities, which are materia alba and calculus. So what is meant by materia alba? Materia alba is a soft accumulation of bacteria and tissue cells which lacks organized structure of dental plaque and can be easily displaced by a water spray. And calculus is a hard deposit formed by mineralization of dental plaque, and it is generally covered by a layer of unmineralized plaque. Dental plaque can be classified into two types, supragingival and subgingival. Supragingival is further categorized into coronal and marginal, and subgingival is categorized into tooth-associated, epithelium-associated, and unattached. So based on the position of plaque on the tooth surface, it can be categorized into supragingival plaque and subgingival plaque. Supragingival plaque is above the gingival margin, and this comes in directly contact with the gingival margin, plays an important role in development of gingivitis. Subgingival plaque is below the gingival margin between the tooth and the gingival sulcus. These are two types, tooth associated and epithelium associated. Tooth associated is plays an important role in calculus formation and root caries, and epithelium associated plays an important role in development of periodontitis. So plaque can be detected into various methods. The first one is direct vision. The second one is use of explorer or probe. And the third one is use of disclosing agents. These includes a two-tone solution, Skinner's solution, Bismarck solution, erythrocin, fast green, fluorescein, and plaque light. The composition of dental plaque consists of microorganisms which are mainly embedded in extracellular matrix. And these microorganisms are mainly bacteria, and the non-bacterial organisms are yeast, mycoplasma, protozoa, and viruses. The host cells in the dental plaque consist of epithelial cells, macrophages, and leukocytes. The intracellular matrix is of two types, organic and inorganic. Organic consists of glycoproteins deriving from saliva, the polysaccharides, which are dextrin and leaven, and proteins, that is albumin, deriving from GCF, and the lipids deriving from bacteria and host cells. Inorganic are calcium, phosphorus, sodium, and potassium, and fluoride. The sources of inorganic constituents, supragingival plaque primarily from saliva, and the subgingival plaque primarily from crevicular fluid. The fluoride component of plaque is largely derived from external sources such as fluoridated toothpaste, rinses, and fluoridated drinking water. The structure of supragingival plaque is mainly covered by a thin layer of acquired pellicle. This consists of mainly gram-negative cocci with epithelial cells and leukocytes adhering to the pellicle. During the first few hours, there is a proliferation of attached bacteria which forms in small colonies. And later, these microorganisms of different species follow the increasing complexity of plaque composition. And subgingival plaque, these are densely packed microbial accumulation, which includes gram-positive and gram-negative cocci and rods, filaments, spirochetes. In periodontal pockets, the surface layer facing the soft tissue is more of spirochetes and flagellated bacteria with gram-negative cocci and rods. In deeper pockets, the number of filaments are decreased. And these subgingival bacteria possess capacity to invade dentinal tubules. These are the various organisms present in supragingival and subgingival plaque. These are gram-positive facultative anaerobes, cocci, and rods. These include Streptococcus species and rods Actinomyces species and anaerobes Streptococcus species and Actinomyces, Naslandi, Viscococcus, and Israeli. Gram-negative facultative anaerobes cocci are Neisseria, and gram-negative anaerobe rods are Actinobacillus, Actinomycetum comitans, Capnocytophaga species, Eikenella species. And gram-negative for cocci, these are anaerobes, Veillonella species, and rods are bacteroid species. Plaque formation occurs in three phases. The first one is pellicle formation, initial adhesion and attachment of microorganisms. And the third one is colonization and plaque maturation. The pellicle, which forms a coating on the tooth surface derived from the saliva, is called the acquired pellicle. This pellicle consists of glycoproteins, fluorine-rich proteins, phosphoproteins, histidine-rich proteins, and enzymes. Studies have indicated that 2R enamel pellicle reveals that acid composition differing from that of saliva indicates that pellicle forms by selective adsorption of environmental macromolecules. And the mechanism involved is electrostatic van der Waals and hydrophobic forces. The ultrastructure of acquired pellicle is thickness is 30 to 100 nanometers. It is a thin basal layer and thicker globular layer. In 2R pellicle formation, it consists of granular structures forming globules connecting to the hydroxypeptide surface via stalk-like structures. In 24 hours, the globular structures will be covered by fibrillar particles, which is of 500 to 900 nanometers thick. So initial adhesion and attachment of plaque on the tooth surface occurs in 4-stage sequence. The first one is transport to surface. The second one is initial adhesion. And the third one is attachment. And the fourth one is colonization of surface and biofilm formation. So initial adhesion and attachment, the phase 1 is transport of bacteria to tooth surface, which mainly occurs by Brownian motion, liquid flow, or active bacterial movement. And the second phase is initial adhesion, resulting in initial reversible adhesion of bacteria when it comes to close proximity with tooth surface through long range or short range forces, which are van der Waals, attractive forces, and electrostatic repulsive forces. And the third phase is attachment. This forms a firm anchorage between the bacteria and the surface, establishing a specific interactions by covalent ionic or hydrogen bonding. The binding between the bacteria and pellicle is mediated by specific adhesins on bacterial surface and complementary receptors in the acquired pellicle. The proteins of acquired pellicle act as receptors for streptococci, for example, alpha amylase, acid proline-rich protein, stadherin, and salivary agglutinin glycoprotein. Other types of attachment are flagella or twitching mobility to type 4 pili, which increase in the number of initial interactions between the bacteria and the bacterial cells and solid surfaces, and to help overcome initial repulsive forces between the bacteria and the surface. And the cell surface proteins of Staphylococcus epidermis plays an important role in initial attachment. Colonization and plaque maturation. These microcolonies form by growth of firmly attached microbes and by the attachment of newly formed bacterial clusters. These initial colonizers attach to the tooth surface, which provides a substrate for secondary colonizers for the attachment. And these secondary colonizers do not directly colonize on the tooth surface, but adhere to the bacteria which is already present in the plaque. So this can be summarized as an immersion of hard non-shedding surface onto oral cavity, adsorption of macromolecules leading to biofilm formation, and the bacterial adhesion to the acquired pellicle formed involving primary plaque formers, which are gram-positive facultative cocci and rods, and subsequent colonization of gram-negative anaerobes. What is meant by co-aggregation? This is first described by Gibson and Nygaard. They have explained that it consists of 18 types of genera from the oral cavity, which has shown some form of co-aggregation. So this is co-aggregation is a highly specific stereochemical interaction of proteins and carbohydrate molecules located on the bacterial cell surface. And these newly accreted cell becomes itself a nascent surface and act as a co-aggregation bridge to the next potential aggregating cell type that passes. It is mediated by lectin-like adhesins and can be inhibited by lactose and other galactosides. These are two types of co-aggregation. The first one is corn-cob formation. Streptococci adhering to the filaments of Bacterionoma or Actinomyces species, and the test tube brush, which is composed of filamentous bacteria, to which gram-negative rods gets attached. Subgingival microbial complex. This is a transition from a healthy oral environment to gingivitis and to periodontal disease triggered by a specific set or complex of bacterial species. These includes early colonizers. These are independent defined complexes, which are Actinomyces nasalandi and Actinomyces viscosus. And the other members are Streptococcus species indicated by yellow color, and the Actinomyces odontolyticus and Veillonella parvula. These are indicated by purple complexes. The second one is secondary colonizers. These are indicated by green color, which is Echinella corrodens, Actinobacillus actinomycetum comitans, and Capnocytophага species. And the orange complexes is Fusobacterium, and the red complex is P. gingivalis, T. forsythii, and T. denticola. So this is a picture explaining about the various types of subgingival microbial complexes depicted in different colors. The orange, red, purple, yellow, green. The red indicates P. gingivalis, T. forsythii, and T. denticola. And the orange one, these consist of P. intermedia, Campylobacter rectus, Campylobacter gracilis, and the yellow color consists of Streptococcus mitis and Streptococcus mitis, Staphylococcus mitis, Staphylococcus oralis, and Streptococcus species. And the purple color consists of V. parvula, and green color consists of C. gingivalis, E. corrodens. The ultrastructural aspect of the dental plaque consists of 3 to 30% of the surface is covered by streptococci in first 2 to 8 hours, followed by a short rapid growth. And after 1 day, this biofilm is fully formed into microbial organization, which occurs in palisade formation and pleomorphism. And there is a marked increase in growth rate, which occurs from 2 to 6 millions. And the plaque thickness gradually increases from 20 to 30 micrometers. The clinical aspect of supragingival plaque formation, in the first 24 hours, there is a negligible growth. That means the bacterial colony needs to reach to a certain level to detect clinically. In the next 3 days, there is a rapid growth. And after 4 days, the 30% of the tooth surface is covered by plaque. And there is a transition from aerobic to anaerobic environment. The factors influencing plaque formation. Initial plaque formation taking place along the gingival margin and from the interdental space is and further extending in coronal direction can be observed. And these plaque formation also starts from the grooves, cracks, and perikymeta or pits. Surface micro roughness like crown, implants, denture bases, there will be more accumulation of plaque and calculus, and the bacterial adhesion is facilitated above a surface micro roughness of 0.2 micrometers. Factors favoring plaque retention are natural factors, calculus, crowding of teeth, enamel projections, and mouth breathing. And unknown factors like overhanging margins of restorations and crowns, and poorly designed clasps and prosthesis saddles. The others are amalgam restorations and microbial factors of saliva, chemical composition of pellicle. Early plaque formation and faster in lower jaw, especially in molar areas and buccal tooth surfaces, interdental areas, and that is more rapid on inflamed gingiva. And peri-implant plaque, this is highly proportion of coccoid cells, lesser number of anaerobes, and low detection of frequencies of periodontal pathogens. Implants with peri-implantitis have high proportion of anaerobic gram-negative rods, motile organisms, and spirochetes. In subgingival plaque formation, studies have shown that subgingival microbiota during first week after mechanical debridement, there is a partial reduction followed by fast regrowth to almost pre-treatment levels within 7 days. So this indicates that there is a high proportion of treated tooth surfaces still harbors plaque and calculus after scaling. And these remaining bacteria were considered primary source for subgingival recolonization. Subgingival microflora was largely dependent on remaining presence of teeth and degree of periodontitis in remaining natural teeth. Physiological properties of dental plaque. There is a transition from gram-positive to gram-negative bacterial plaque development. Early colonizers use oxygen and reduce the reduction potential, further favoring anaerobic bacterial growth. And these gram-positive species use sugar as energy source and saliva as carbon source. The byproducts formed from these early colonizers are used in the metabolism of other microorganisms. For example, growth of P. gingivalis is enhanced by byproducts produced from other organisms like C. ochraceus, which is succinate, and C. rectus, which is protoheme. Heme and iron from host hemoglobin is important in metabolism of P. gingivalis. The three theories for formation of plaque and which helps in resulting in periodontal disease are non-specific plaque hypothesis, specific plaque hypothesis, and ecological plaque hypothesis. Non-specific plaque hypothesis was described by W.D. Miller. In this hypothesis, it has been explained that small amount of plaque are present and these noxious products are neutralized by the host, and the large amount of plaque would produce large amount of noxious products, which usually overwhelm the host defenses. So in this concept, it has been explained that the control of periodontal disease depends on control of amount of plaque accumulation. And the treatment of periodontitis by debridement by surgical or non-surgical and oral hygiene measures focuses on removal of plaque and its products and is founded in the non-specific plaque hypothesis. The observation of this hypothesis was that considerable amount of plaque and calculus and with gingivitis never developed destructive periodontitis. And the individuals with periodontitis showed site specificity in disease pattern. And these sites remained unaffected, whereas some sites showed advanced diseases. So the second hypothesis is specific plaque hypothesis explained by Loesch in 1976. And according to this theory, plaque harboring specific bacterial pathogens resulting in periodontal disease because these organisms produce substances that mediate the destruction of host tissues. And the third one is ecological plaque hypothesis explained by Marsh and Bradshaw. According to this theory, the disease resulting from the shift in the balance of resident plaque microflora, providing favorable conditions for growth of pathogenic organisms or expression of pathogenic traits. So there is a change in the nutrition status of a pocket or chemical and physical changes of the habitat and considering the primary cause for the overgrowth by the pathogens. So according to this theory, the disease could be controlled not only by targeting pathogens, but also by interfering with the factors responsible for driving the deleterious shifts in the microflora by reducing the GCF rate, or the site could be made less anaerobic by use of redox agents. What is meant by biofilm and why the biofilm is called as plaque? Biofilm is referred as a relatively undefinable microbial community associated with the tooth surface or any other hard non-shredding material randomly distributed in a shaped matrix or glycocalyx. And this consists of three layers: lower layer, loose layer, and fluid medium. Loose layer consists of dense microbes which are bound together in a polysaccharide matrix with other organic and inorganic materials, and loose layer is a loose irregular layer extending into the surrounding medium, and this consists of a fluid medium bordering the biofilm. Properties of biofilm. Microorganisms are arranged in microcolonies with channels, and these microcolonies are surrounded by protective matrix and thus differing environments within the microbiofilms in the biofilm. Biofilm microbial gene expression differs when microorganisms are in a biofilm. The microorganisms and biofilm are very resistant to antibiotics, antimicrobials, and host response. Dental plaque represents a true biofilm which consists of bacteria in extracellular matrix composed of bacterial polymers and saliva or gingival exudate products. The further discussion will be explained in the next class.